All right, so let's jump right into operons. So as already mentioned, there's some genes that are constitutively on. They're on all the time for structural proteins, some enzymes that need to be functioning at all times. So these genes are going to be on all the time. Other genes are only going to be expressed as needed. So they can be turned on and off based on whether the protein for that gene is needed at the time. There's two types. There's repression, which is going to involve which is going to inhibit or repress gene expression. And this is mediated by repressors, which are proteins that will block transcription. Now, these genes are going to be on. Their default position is on, and then they have to be repressed to be turned off. The other type are induction. So these genes are going to be normally turned off, and then they're going to use an inducer, another type of protein, to turn it on or induce that gene on. So there's a few parts to the operon. The operon includes the operator and the promoter and the structural genes that they control. So we've already talked about the promoter. That's usually the first segment. So the promoter is like the on-ramp onto the freeway. It's telling the RNA polymerase, this is the start of a new gene, come here. So it's calling the RNA polymerase to the right starting position. Then the next segment of the operon is the operator. The operator is kind of like the light when you first get on the on-ramp to the freeway that's saying stop or go. So that's going, you're already ready to get on the freeway. So the RNA polymerase is already here at the start, but the operator is controlling whether it's going to continue or not. So the operator is what's turning that gene on or off. And then we have the structural genes. So one more time, this whole thing here is the operon. We have the promoter, the operator, this is the control area where we're turning it on or off, and then the structural genes. So these are the actual genes that are going to turn into proteins, or that encode for proteins, rather. Now, why do we see operons so much? Well, they're very strongly favored by natural selection. So we see this in a lot of different species. We see that it's evolutionarily conserved. So we see it amongst all these different species. We don't see operons removed a lot by natural selection. And that's because they are saving so much energy. Again, there's no point in having a gene turned on to produce proteins that aren't needed. So that's why operons, we see them a lot for all of these genes that aren't needed all the time. All right, so our first example is an inducible operon. So here, under normal conditions, the gene is going to be turned off, and it's turned off by this repressor that is normally active. So when the active repressor is bound to the operator, the RNA polymerase is blocked. It cannot go and transcribe these structural genes. Now, this is the Lac Operon. We call this the Lac Operon because it's encoding for lots of enzymes that are needed to break down lactose. So without lactose, why would you have enzymes to break down lactose, right? So that gene is going to be turned off when we don't have lactose. Makes sense, right? So now we have the presence of lactose. Lactose is what we're going to call the inducer. And that is going to bind to the repressor. This repressor is always on unless it's bound to lactose, and that will inactivate it. So now our repressor is inactivated. An inactivated repressor is no longer going to bind to the operator. Now with nothing bound here, the RNA polymerase is free to continue on and transcribe these Lac genes, which will then produce these enzymes that can break down lactose. Now, I wish it was that simple, but it gets even more complicated. So what we just talked about is inducing or turning this gene on when we have lactose. Because why would we have enzymes to break down lactose if we don't have lactose, right? Now, to make the story a little bit more complicated, bacterial cells always have enzymes needed to break down glucose. So it's going to prefer to break down glucose if that is present. It's also more efficient for the cell. So if we have glucose, the cell would rather spend the energy in breaking down and metabolizing glucose rather than spending energy to produce the enzymes needed to break down lactose. So in order to ensure that it's going to metabolize all available glucose first, we have this second part. First, let's note that CAMP, this is a molecule here, is inversely related to the presence of glucose. So if we have a lot of glucose, we're not going to have CAMP. If we don't have a lot of glucose, we're going to have CAMP. So in this first part, we have CAMP, which means that we do not have a lot of glucose. Glucose is scarce. We also see that lactose is present. So we do have lactose, but we don't have glucose. So now it's ideal for the cell to start producing the enzymes needed to break down lactose. So it's going to, the CAMP is going to bind to this protein, CAP, which is normally inactive. Once it has been bound to CAMP, it's now active. So now we have an active CAP. What an active CAP does is it's going to increase the affinity of RNA polymerase for the promoter. So it's like putting on bright lights to the freeway on-ramp, like even brighter than it already was, telling it, all right, let's go, right? Let's start transcribing this gene right here. So again, without glucose and with lactose, we're going to have CAMP that's activating the CAP, which increases the affinity to RNA polymerase. And because we had lactose and that repressor is inactive, RNA polymerase is free to transcribe these genes to break down lactose. Right, so then in this case, we do have lactose, so we could potentially metabolize lactose, but we do also have glucose. So the cell would rather metabolize glucose. So in this case, we don't have CAMP because with glucose, we're not going to have CAMP, remember? So no CAMP means we're not going to activate the CAP, which means we have a lower affinity for RNA polymerase. So it's less likely to bind and transcribe these genes. It could, because we don't have the repressor bound because we have lactose, but it's less likely to occur. So these graphs are just showing the differences between glucose and lactose metabolism. So in the presence of either glucose or lactose, we see that glucose has a higher growth rate. So the cells grow faster when they're metabolizing glucose than they do with lactose, which is why they would prefer to utilize glucose and lactose. Now, if the cells are grown in the presence of both glucose and lactose, what we see is a faster growth rate initially until all the glucose has been utilized, and then the cells will switch over by turning on this Lac Operon to start metabolizing lactose, which is a slightly slower growth rate, but growth nonetheless. So that was an inducible operon. Now looking at a repressible operon, we're going to look at tryptophan. So this is repressible. What we're seeing here is this operon is encoding for several enzymes that are needed in the metabolic pathway to produce tryptophan. Now, tryptophan itself can regulate its own production in two ways. Tryptophan can serve through feedback inhibition to repress this enzyme, the first enzyme in the pathway. Or tryptophan can regulate gene expression. And that's what we're going to look at here. We've already looked at feedback inhibition in an earlier chapter. And now we're going to look at the regulation of gene expression. So being that this is a repressible operon, it's going to normally be on. So in this case, our repressor is inactive. So our operator does not have the repressor bound. RNA polymerase is free to transcribe these genes to encode the enzymes that will produce tryptophan. So our gene is currently on. This is going to be occurring when tryptophan is scarce, right? We're making the enzymes that are going to produce tryptophan, which is needed since it's scarce. However, all right, sorry. However, when we have plenty of tryptophan, then we're going to turn that gene off. And that's going to happen by tryptophan itself is going to act as a co-repressor. So the tryptophan is going to bind to the inactive repressor, which will then activate it. Now that the repressor is activated, it's going to bind to the operator, which will block RNA polymerase from transcribing these genes. So we're no longer going to make the enzymes that are needed to produce tryptophan. Why would we? We don't need any more tryptophan. There's already plenty of it. So that's our repressible operon. Yay, we made it through operons. I know that may seem like a lot of information. So if you need to, feel free to pause and rewatch that part on operons. If you are still with me, great. Let's continue on. We're going to talk about mutations next. So mutations are, of course, changes to the DNA sequence, and that's going to increase the genetic variation amongst these different cells. Mutations can be either beneficial, harmful, or neutral. An example of a beneficial mutation would be antibiotic resistance. This is beneficial for the microbe. Other mutations can cause proteins that are needed to no longer be functional, and that would be an example of a harmful mutation. Or some mutations can just be neutral. They don't have any effect on the microbe's ability to survive and reproduce. There's several types of mutations that you need to be familiar with. So the first one is missense mutation here. This is just going to trade out one nucleotide for another. So whereas this nucleotide it's just changing it out. So where this should have been, let's see, we have AAACC, but now we have AAATC. So this is our mutation here. So that's going to, of course, affect the mRNA and affect the amino acid that's then translated. A nonsense mutation is going to encode for a stop codon early. So whereas, see, this is our normal over here, we should have all these different amino acids that are translated. Instead, this is now going to become a stop here. And so we don't have all these other amino acids produced. And the third one that I want you guys to know is the frame shift mutation. So in this case, we're either going to be removing or adding in an extra nucleotide. In this case, a nucleotide was removed. And so what that does is that shifts all the downstream nucleotides over, which is going to change the codons, all the rest of the codons over because everything was shifted. So whereas we used to have, let's see, CCG, oh no, here, we had AAA, right? Well, now we have AAC. And so that's going to change this amino acid. But instead of having CCG, now we have CGA because everything was shifted over. So that's going to change this amino acid. It's going to change all the rest of the amino acids downstream. So nonsense and frame shift mutations can be quite detrimental because it's affecting not only that amino acid, but all of the rest as well. Whereas a missense mutation is simply going to change just where the nucleotide mutation occurred. Looking at genetic recombination. So this is where genes are exchanged between two DNA molecules. This can increase genetic diversity and is carried out through the process of crossing over. Don't worry too much about the Rec A protein or anything. I just want you to be aware of what genetic recombination is and what crossing over is. This should sound familiar because this is the same process that occurs during meiosis, right? When we're the genetic recombination of the parental genes crossing over. This can also happen in bacteria. Remember that vertical gene transfer is the passing of chromosomal traits from parent to offspring. So parent cell to offspring, the daughter cell, that would be vertical gene transfer. We talked about that earlier when we talked about cell replication or DNA replication. Now we're going to talk about horizontal gene transfer. So now we're talking about transferring genes from one cell to another cell within the same generation. And that can happen through three main ways. It can either happen through transformation, conjugation, or transduction. Transformation is where DNA, or we call it naked DNA, that's found in the environment is taken up into a bacterial cell. So there can be just DNA fragments for whatever reason that's just found in the environment of the cell. Then that cell will take it up and that it can be incorporated into its chromosome. The process of transformation was proved through Griffith's experiments. So what he did is first he injected a mouse with living encapsulated bacteria. So these are a pathogenic species, and that ends up killing the mouse, and then he's able to culture the bacterial species from the dead mouse, and he sees that they're encapsulated just like they were before. Then he takes another species, they're living, non-encapsulated bacteria. Without the capsule, they are able to be overcome by the mouse's immune system, so he does not succumb to the disease, but Griffith is still able to culture those bacteria, and they didn't change. They're still living, non-encapsulated bacteria. Then he takes the initial species from over here, and he kills them. So they're still encapsulated, but they're killed. Injected into the mouse. Well, they don't make him sick because those bacteria are dead. And we don't see any bacteria from the culture because they didn't grow because they were dead. And then in the last step, he combined the two. So he injected the mouse with both living, non-encapsulated, so the non-encapsulated, less pathogenic species was living, and he injected the mouse with heat-killed encapsulated bacteria. So he injected them both into the mouse. Now remember, both of these did not kill the mouse. In both of these, the mouse was fine. But in this case, when he injected them both, the mouse died. And then when he cultured the bacteria from the dead mouse, he found living encapsulated bacteria. Well, we know that these bacteria would be coming from these unencapsulated or non-encapsulated bacterial species, the less pathogenic one, because they're the only ones that were alive. They're the only ones that could have infected the mouse. But they are now encapsulated, so they must have gotten the genetic information in order to become encapsulated from the dead bacteria. So the encapsulated dead bacteria was able to release DNA fragments into the environment that were taken up by the living unencapsulated bacteria, which then were able to incorporate that pathogenicity, and then we're able to become pathogenic and kill the mouse. The second way is conjugation. So conjugation is where plasmids or chromosomal DNA are transferred through direct cell-to-cell contact using this cell pelus. So this is like a hollow tube here. It's going to transfer plasmids most commonly, but it can also transfer chromosomal DNA. And the third one is transduction. This occurs through a bacteriophage. So a bacteriophage is a virus that infects bacteria. So we haven't talked about viruses yet, but in essence, what happens when a virus replicates is it injects its DNA into the donor cell. It's going to replicate that genetic information that's going to be repackaged up into new viral protein coats, which will then go and infect a new cell. But what can happen through transduction is when the viral genes are packaged up into its protein coat, it can accidentally package up bacterial DNA instead. And then unknowingly, that virion will then go and inject those genes into a new bacteria, which will then incorporate that bacterial DNA into its genome. So we just talked about those three different main methods, but before we conclude, I just want to touch on these mobile genetic elements. So there's these different genetic elements that can move either from one chromosome to another within the same cell, or it can move from one cell to another. And we've already talked about plasmids. It's an example of a mobile genetic element. We know that plasmids is a small circular loop of DNA containing genes, most commonly like antibiotic resistance, or other genes that can increase the pathogenicity, increase virulence, can encode for different toxins, etc. One last example I'm going to talk about is transposons. This is an area of research currently, but these are small linear DNA segments that can move from one chromosome to another. So they can move around, which means they can move from the viral DNA to plasmids to the main chromosome, and they move around within the cell and to other cells, so they can move genes around. So we're going to end on that. And that's chapter 8. That's going to conclude everything for our unit 2. So we have chapter 5, 6, and 8 that will all be on exam 2. As always, please feel free to email me or ask me any questions. See you guys soon.